Hello and welcome back to second video of instrumentation from Fluorometry. We will continue from previous video. In the previous video, we have learned about the theory, theory of the fluorometry determination. Now in this video, we'll start with the instrumentation. There are two type of instrument used in fluorometric measurement. One is your filter fluorimeter, another one is your spectropluorimeter. The basic component of any instrument include source of light, filters and monochromators, sample cell or sample holder, detector and readout unit. This is the block diagram for any instrument, either it is a filter fluorimeter or spectropluorimeter. The first component is your light source. Generally, for fluorometric determination, we are using the line sources. We have studied two types of sources in previous lecture. One is called as a line source, which gives the discrete lines of the radiation. Another one is a continuous source, which gives the range of radiation within the wavelength region. Now here we are using sources of the single wavelength. Therefore, they are called as line sources. Then this again the line sources, we need some excitation monochromators or excitation filters. Then this is the slit, then sample cell. To the right angle to the source, the another filter is situated, that is called as the emission filter or emission monochromator, where the emitted radiations are filtered and they are sent to the detector. And this after detector, there are the recorder and amplifier called as a readout unit. So this is the block diagram. Here you have to remember that the source is 90° to the detector. Why? That you have to think. Now we'll start with the source. So mercury arc lamp we have studied in the UV visible spectroscopy also. The second is one is xenon arc lamp. Third one is a tungsten filament lamp, and fourth one is a tunable laser lamp. So these are the various sources available. You can see the picture of mercury vapor lamp, then tungsten filament lamp, and your second one is a xenon arc lamp. The tunable lasers. Do you know what is the long form of laser? LASER. Yes, light amplification by stimulated emission of radiations. Again, I will repeat, light amplification by stimulated emission of radiations. Any light can be converted to the laser beam. Either you can convert visible light. So you have seen this visible light used in various programs where the focus light, you call it as a focus lasers, are used. UV light is used for some eye operation purpose. Laser beams of the UV lights are used. So this is a tunable carbon dioxide laser source, okay, where we can adjust the um, wavelength from 250 to 2500 nanometer. So this is a tunable laser source. So these are the sources. Out of this, first is your mercury vapor lamp. At high pressure gives intense lines on continuous background above 250 nanometer. So here low pressure mercury vapor gives an additional lines at 254 nanometer. It is used in filter fluorometer where you require the single wavelength. Then second one is a xenon arc lamp. It gives more intense radiation than mercury vapor lamp. It is used in spectropluorimeter because xenon arc lamp can give all wavelength from 200 to 400 nanometers of UV and 400 to 1000 nanometer of the visible and some near IR region. So xenon arc lamp is a only one source which can you use in UV and visible region. The next one is a tungsten filament lamp. It is used in the visible region. It is excitation is to be done in visible region. It is used in low cost instrument where we are using the filter fluorometers as a low cost instrument. Then second component is filters and monochromators. If you are using filter, these are nothing but the optical filters working on the principle of absorption of unwanted light and transmitting the desired required wavelength of light to the other part. We have studied this absorption filter. They are the colored glasses or colored dyes which are imbibed between the two transparent plates. Okay. Again, here also we can use the interference filter. Interference filter, which is having the mercury, calcium fluoride, magnesium fluoride, like the material which is sandwiched between the two transparent plates. In all fluorometry, primary filters and secondary filters are present. Primary filter is known as a excitation filter, and secondary filter is known as the emission filter. Primary filter absorbs visible radiation and transmit UV radiation, whereas the secondary filter absorbs UV radiation and transmits the visible radiations. Okay, so when we are bombarding UV light, you are getting the visible light out. So you may have seen such type of the filters in your instrument with different types of the wavelengths can be utilized for all determination. Monochromators. Again, the principle of monochromator is same as we have seen in the previous. There are either prism monochromators or grating monochromators. Okay, so in translate, then collimating lens, collimating mirror, then dispersing devices like gratings or prisms, then focusing lens, focusing mirror, and exit slits. These are the five basic components of any monochromators. We have already seen the working basic principle of monochromator. Just by rotating the dispersing devices, you may get the different wavelength out. They converts a polychromatic light into the monochromatic light. They can isolate a specific range of wavelength or a particular wavelength of radiation from the source. There are also two types of monochromators used. First one is your excitation monochromator, which provide suitable radiations for excitation of molecule, and second one is your emission monochromator, which isolate only the radiation emitted by the fluorescent molecule. Now these are meant for sample cells. Okay, so these are meant for holding the liquid sample. These are made up of the quartz. Okay, can give various shape. Example, cylindrical or rectangular. Okay, so these are the cylindrical sample cells, and these are the rectangular sample cells. One thing difference you can observe in the cuvettes which are used in fluorometry and cuvettes which are used in spectroscopy. What difference you are observing in this sea? There are no ground surface. All four sides are transparent. Okay. Here also circular, all four sides, all sides are transparent. There is no ground surface because at the 90 angle we are getting the re-emitted radiation. We are not measuring the transmitted radiation. The last component is detector. Okay, so detectors we have studied in the UV visible spectroscopy. Photometric detectors are used here, such as a barrier layer cell, that is called as a photovoltaic cell. Then phototubes, that is modified form of phototubes, is a photomultiplier cell which work at the 90 electrovolts. It has a 9 diodes. At each diodes there is a multiplication of cascade, multiplication of electron produced. So at last it produces a cascade of electron. So it will give us a intense output. So these are the sample holder. So the instrument most commonly used are of two types: single beam filter fluorometer or double beam filter fluorometers or spectrofluorometer. They are generally double beam instruments only. See the construction of single beam instrument. Single beam means only one beam continuously passes from the sample and reference. You have to replace the sample and the reference cavity within the path. Okay, light source, single cavity is there, and the detector. But in case of the double beam, there is a beam splitter which will split the light in two beams. One beam is continuously passing through the sample, another beam is passing through the reference. Here, don't consider reference as some other substance. Reference are generally the solvents, because we don't want any absorption or fluorescence of sample. So effect of sample is to be nullified by using this double beam instruments. Okay, so then again there is a combiner, or we can have the two detector systems also. So this is a single beam filter fluorometer or double beam filter fluorometer. The another one is a spectrofluorometer. In case of spectrofluorometer, there are two monochromators used. One is known as the emission monochromator, another is known as the excitation monochromator. So here we are using the xenon lamp as a source. So the radiation from xenon lamp passes into the first monochromator, where its fixed wavelength will be bombarded. It will be splitted into the two part. One part is going towards your reference and another part is going to your sample compartment. Then the radiation which is reflected through the sample and reference are passed to the another monochromator, where the emission spectra will be detected. So whatever light absorbed, it will goes into the excited state, it will remit back the radiations, and these emitted radiations are further filtered by using your excitation monochromator, emission monochromator, and then they will be sent to a sample photomultiplier tube. Okay, so we can use the photomultiplier tube as your detector. So single beam instruments, it contains tungsten lamp as a source of light and has an optical system consisting of primary filter and secondary filter. The emitted radiation is measured at 90 degree by using a secondary filter and detector. Okay, so you have to remember why 90 degree. Primary filter absorb visible radiation and transmit UV radiation which excites the molecule present in the sample. Instead of 90, if we use 180 geometry as a colorimeter, secondary filter has to be highly efficient. Otherwise, both the unabsorbed UV radiation and fluorescent radiation will produce detector response, unwanted detector response, and gives faulty results. Okay, so we cannot keep it into the straight line. It is always to be kept into the 90 degree. So this is always asked. The single beam instruments are simple in construction, cheaper and easy to operate. The double beam instrument, it is similar to the single beam instrument except that the two incident beams from the single light source passes through the primary filter separately and falls onto the another reference solution. Then the emitted radiation from the sample or the reference sample passes separately. Okay, see here. So they are passing separately from the sample and your um, reference cell through the secondary filter and produce a response combinedly on a detector. In a spectrophlorometer, the primary filter is a double beam fluorometer is replaced by excitation monochromator and secondary filter is replaced by emission monochromator. Incident beam is split into the sample and the reference beam by using a beam splitter. Now we will go to the applications of your fluorometer. For pharmaceutical analysis, many times we are using it a very sensitive tool because very small concentration. So you have to find out the advantages and disadvantages of your fluorometry. So advantages, I can say that it is a very sensitive. Sensitive means very small quantity of the samples can be analyzed. Secondly, the advantage that it can be used for mixture of substances. Even if you have the multi-component formulations, all multi-component drugs can be analyzed by keeping together simultaneously because every substance has its own excitation wavelength and own emission wavelength. So they will not interfere in determination. So fluorometry is having this advantage. But what is the limitation? Because it is a very sensitive technique, very small concentration of the substances can be determined. We require the various proper adjustment of the solution, that their pH, dissolved oxygen. Okay, so this all material or the concentration, this will produce the effect on the determination. So advantages and limitations are there because all substance will not produce the fluorescence. We need the substance which shows the fluorescence. Okay, there should be the double bond system or there should be the non-bonded electron, then and then only it will show the fluorescence. Every substance is not producing fluorescence, even the presence of non-bonded electron. Then what we can do, many substances can be converted to the fluorogenic derivatives like your thymine. Thymine hydrochloride is non-fluorescence, but it can be converted to the fluorogenic derivative, that is thiochrome, okay, by oxidation treatment. Similarly, your adrenaline is also non-fluorogenic. It can be converted to the adenone, which is having the fluorescent nature. So it is used for the determination of B1 and B2. B1, that is thymine hydrochloride. B2 that is ribofulvin, B6 and B12 that is cyanocobalamin. Determination of thiamine hydrochloride, determination of phenytoin, determination of indoles, phenols and phenothiazines, determination of naphthals, protein, plant pigments and steroid. In inorganic substances determination, it is also used for determination of ruthenium ions in presence of other platinum metal, determination of aluminium in alloys, Al3+, determination of boron in steel by complex formation with benzene, and estimation of cadmium with 2-hydroxyphenyl benzoxole in presence of tartarate, and in field determination of uranium salt. It is also used as a fluorescent indicator in the acid-base titration. Example, we have seen eosine, which can changes its color from colorless to green. Then fluorescein, colorless to green. Eosine, fluorescein. Then quinine sulfate, blue-violet. Acridine, acridine, green to violet. So these are used as a indicators in some titrations. So some examples with their filters, excitation filters and emission filters are listed here, which are commonly asked. We are doing the quinine sulfate determination in laboratory. So we have to work in with the 0.05 molar H2SO4. So that is within the pH range of 1. So excitation wavelength is 350 and your emission wavelength is 475. Same is for the thiamine hydrochloride. Means we can use the same filter for quinine sulfate and thiamine hydrochloride, but if you are going to use the ribofulvin as another substance, we have to use the 470 nanometer and 520 nanometer as a filters, primary filter and secondary filter. In cyanocobalamin, we have to use 250 nanometer as a excitation wavelength and 370 nanometer as your emission wavelength. Morphine can be determined in same way. The pH is 7. In neutral pH, we have to determine morphine. Then phenobarbital, we have to determine in the alkaline pH, and hydroxycortisone, we have to determine in the acidic pH. So here we have completed the another topic, that is fluorometry as per your syllabus. Phosphorometry is not covered in your syllabus, so we will not focus on some part of the phosphorometry applications or instrumentation, etc. Thank you and happy learning, friend. See you in the next video. Solve all the questions and quiz after this lecture. Thank you.